the problem that was arise in the last slide is rectified by analyzing the two proteins that are very similar. So, for example, we can take the two proteins that are only different in one amino acid. That is, in out of 100 amino acids, there is one amino acid that is different in the two proteins, or either one indel is present between the two similar proteins. So such sequences, they are present within the humans and chimpanzees, and these protein sequence, they fulfill the criteria of 1% indels or mismatches in 100 amino acids. So these similar proteins that are only 1% different with each other, they are generally recognized as 1 PAM unit diverged. So 1 PAM unit is defined as the amount of time in which the average protein mutates to only 1% of its amino acids. And PAM1 scoring matrix, because PAM1 means the PAM 1% in the scoring matrix in which there is a 1% difference in the amino acids. So PAM1 scoring matrix is defined as many alignments of extreme similar proteins, and it is defined as follows in the next slide. So we will see how PAM1 scoring matrix is defined. It is defined that it's for a given set of base alignments. Now we will define various parameters. For example, first is we will define Fij, that is the total number of times amino acids I and J are aligned against each other. It means that the number of times amino acid I in V string and the J amino acid in the W strings, they are identical at the same positions, and these number of amino acids will be divided by the total number of aligned positions. So how many positions at which these amino acids are identical? It will be divided by the total number of amino acids. Second is we will define the Gij as the Fij divided by Fi, where Fi is the frequency of the amino acid I in all proteins from this data set. And the third is Gij defines the probability that an amino acid I mutates into the amino acid J within 1 PAM unit. We have defined the 1 PAM unit as a time in which the one amino acid is mutated out of 100. So the Ij entry of the PAM1 matrix is defined as delta Ij is equal to log Fij divided by Fi Fj, and that is equal to Gij F divided by Fj. In this case, we have replaced the value of this one, Fij divided by Fi, and if we replace this value with Gij, then we will left with Gij divided by this Fj. So in this case, we have replaced the value of Fij divided by Fi with Gij, because Gij is this value. And in this Fi and Fj, these are the frequency of aligning amino acid I against the amino acid J. We already seen previously, and that one expect is simply by chance, because there's a mutation and they can change replace with each other. So the PAM-N matrix can be defined as a result of applying PAM1 I1 matrix n times. And if G is, we have already defined the G, that is 20 into 20, because we have dealing with the amino acids, proteins, and in protein there are 20 amino acids. So 20 into 20 matrix of frequencies Gij, then Gn, that is the multiplication this matrix by itself by n times. That gives the probability that amino acid I mutates into amino acid J in n PAM units. So very simple, then Ij entry in PAM n will be replaced by this G with Gn, and we will come up with the, because in the last slide we see we have replaced Gij. Now we will replace G with the Gn here. So we will replace G with Gn and we will divide it with Fj. So for the large n, the resulting PAM matrices often allow one of to find related proteins even when the particularly no matches in the alignment. So in this case, the underlying nucleotide sequence are so diverse that their comparison usually fails to find any statistical significant similarities. So this is more suitable for amino acids rather than the nucleotide sequences. So for example, if we consider the two genes and we see the similarity between these two genes, for example, here we consider the cancer causing V6 oncogene, and if we compare it with another gene, that is the growth factor PGDF, means the platelet derived growth factor, and this PDGF is involved in the cell division and cell growth. So this growth factor, if we compare with the cancer causing gene, then this alignment will remain undetected. So this problem is solved by the scientists Russell Doolittle and its colleagues, and what they have do is they have transformed the nucleotide sequence into the amino acid sequences, and they have done this prior to performing the comparison, because this type of sequence and the scoring matrix is not suitable for the genes. It means they are not suitable for the nucleotides. So they have converted and transformed these nucleotides into the amino acids before performing this comparison.